Welcome back to the On Helix digital live stream for the Leveraging All of Life Sciences for a Better Healthcare Future session. So as we heard several times during the course of the conference, and actually reinforced this morning with Ian Charles' presentation, improving healthcare or actually preventing the next healthcare crisis cannot only come from developing therapeutics, whether it's about having synthetic biology approaches to replace chemical use, or broader solutions with positive impact on the environment. These are not only doing obvious good for healthcare, but they're also developing technology that can translate in the therapeutics life science space. So we are now going to hear three great examples of these. As usual, you'll be able to ask them questions via the chat box on the event app. And as we couldn't run a session like that without a great chair, I'm delighted to invite Nadia Shivji-Brown from Public Health England to moderate it. So Nadia, over to you.
Thanks, Leanne, and thanks for the invitation to chair the session and to One Nucleus for organising another great On Helix. So as Leanne said, we're welcome to the Leveraging of All Life Sciences for a Better Healthcare Future session. We've got three great companies, they coming from Norwich and Cambridge. As Leanne said, if you've got any questions, put it in the chat box, we'll pick them up and we'll be taking questions after each speaker. So first, I don't want to take up their time, so I'll move on to Leanne Williams. She's Business Development Manager for Leaf Expression Systems, and she'll be talking about their protein expression system. So hand over to you, Leanne.
Hi, thanks, Nadia. Hi, can I have my slides, please? Great, thank you. So yeah, I'm Leanne and I work for Leaf Expression Systems. Next slide, please. So just a little background about Leaf. I just call us Leaf from now on because we're quite a long name. So we were incorporated in 2016. It was spun out of a lab in the John Innes Centre in Norwich. So we had our purpose-built facility, which is on the Norwich Research Park, opened in 2017 by the Science Minister, Joe Johnson. And then we became fully operational with our first commercial client project in 2019. And then again in 2019, we launched our first line of products using our plant-based expression system. We're not a catalogue company, we are a CDMO, but we do have a range of catalogue products which are to demonstrate what we can do with our technology. Next slide, please. Right, so at Leaf, we are experts in plant-based expression, and our mission is to improve lives by accelerating clients' research and lowering the costs of development, manufacture of therapeutic drugs, vaccines, and diagnostics. So, as I said before, we were spun out with the John Innes Centre. This is George Lomonosov's lab and Frank Sainsbury, which the hypertrans system we use, plant-based expression system, was awarded the BBSRC Innovator of the Year in 2012. In addition to hypertrans, we're continually developing our own expression technologies in order to advance efficiencies and yields. We've actually just filed a patent for our new technology, which is coming online for our clients right now. We employ the non-cultivated tobacco species, Nicotiana benthamiana, to produce our proteins in. And our system is very rapid, cost-effective, and highly scalable, and as well as being sustainable. Next slide, please. So you might all be thinking, why plants? Compared to the conventional systems that people would use for protein expression. So we have projected lower capital costs for the plants. Our system is very, very fast. So because we do transient expression within our system, so we can get from getting a sequence of a protein all the way to producing that actual protein in 16 weeks, which is a lot faster than traditional bioreactor systems. The system is very easy to scale up. So we don't have to worry about media and concentrations of various things that would go into a giant bioreactor. We just grow more plants, and as those plants are grown in the same conditions, they behave much the same way, produce the same yields. So it's very straightforward then to just grow more plants. There's a low risk of contamination because it is an animal-free system. So you don't have the contaminants that you would have in a normal system that might be harmful to human health. The system is very simple for generating high biomasses. Again, bigger growth rooms, more plants, more biomass. Again, expression, toxic proteins work quite well in our system. Again, because it's transient, the plant doesn't really have times quite often to work out that the protein is actually toxic. So it'll only express the protein for approximately 10 days. And within that time, we can take the best time point and purify from there. In addition to this, plants are fully eukaryotic systems, so they produce a wide range of post-translational modifications. So you get a complex functional protein produced from our system. So next slide, please. So how do we do this? So we use the bacteria Agrobacterium tumefaciens. So Agrobacterium naturally transforms the plants. So the DNA is transferred from the plasmid in the hypertrans system into the plant genetic material, and then the transformation occurs. This can be done both stably and transiently, but stable transformants, they take a long time. You're looking at 6 to 9 months minimum to produce a stable transformant. And also you have issues of silencing down the line when you produce generations of plants. Transient events also at a much higher frequency. So this is what we use for our system. We use a transient-based expression system. Next slide, please. So this is how we do it. So we take the Nicotiana benthamiana plants, and we have a vacuum infiltration system. So we'll grow up a large volume of bacteria with the gene of interest in it. The plants get inverted in this bacterial solution, and a vacuum means the plants take up this agrobacterium into the spaces in between the cells. And then the plant uses these new instructions from the agrobacterium to produce the protein of interest, which will build up inside the plants. Then we'll harvest the leaves at the right time point. We'll usually do a testing to see which is the best time point for which protein. And then the leaves are harvested, blended into a liquid. We'll separate the solid mass from the lysates. And then our downstream process is much the same as a conventional system. We will go through a series of purification techniques, which are all chromatography based, depending on the protein of interest. So you could use size exclusion or anion exchange, it depends. And we tailor that for each protein. And then you get purified protein at the end of the purification process. Next slide, please. So the advantages of the system is that it's very fast. As I said before, it's 16 weeks from initial gene sequence to producing a protein. The cost is low and directly scalable, free from animal products, so reducing the threat of contaminations, complete eukaryotic processing machinery, including complex post-translational modifications, so glycosylation, phosphorylation, and amongst many other modifications. So the good thing about our system is that within the hypertrans system that we use for expression, we don't have any size issues with putting proteins in. We can put any gene of any size in. So we quite often get big problematic genes that don't do well in other systems. And again, another great advantage of the system is that it's very sustainable. So our waste is minimal. We reuse all our, we don't use single-use plastics, we reuse all our plant growing materials, we autoclave things, and our plant material, because it is a transient expression system, we can autoclave that, and then that all gets turned into compost. So it's a very sustainable system, as well as being very cost effective and productive. So next slide, please. So I'd just like to give you a brief overview. There's not a lot of time to go through this entire slide, but the timescales, if you were to go, this is the timescales we've projected for a monoclonal antibody manufacture, mammalian traditional bioreactor system versus our plant-based system. So the upstream development time is a lot shorter. We can overlap times much earlier on in the system. So once we've started expressing a protein, then we can go straight down to downstream development. So that kind of the overlap speeds things up a lot. The scale up is straightforward. As I said earlier, more plants just grow more plants and they produce more protein. There's also less start-stop decision-making points along the way. So that sort of stops, that keeps the flow going in the whole system, so it's more smooth. So this basically saves 7 months in the whole manufacturing process, which not only means that you are getting products out to the market faster, therefore improving lives and saving lives. You also have the benefit of having a patent life extended, which is very important when you're looking at pharmaceutical products. So next slide, please. I'd just like to give you a few little examples of case studies we've done, just to show that this works and we can produce these products. So next slide, please. Next, sorry. So this is just an example of a monoclonal antibody that we have produced, a Avastin antibody, and some functional characterization has been done with it. So in vitro binding assays to its native ligand, VEGF-A, which we outsourced for somebody else to do. And this showed our plant-based antibody showed exactly the same behavior as the original bioreactor-based antibody, which is great for the system, shows that it works well. Next slide, please. So again, with this functional characterization we did luminescent cell-based activity assay blockades of the VEGF signal, and this, as you can see from the graph, the leaf antibody that we produce in our plant-based system is not any different behavior-wise from the Biorad human anti-VEGF, which is the original bioreactor monoclonal antibody produced. So there's identical cell-based activity assay profile there. So it's good to know that our products are comparable with traditional methods. Next slide, please. So this is an antibody. I can't tell you what antibody it was, unfortunately, because it's a customer product. But this is an antibody we produced for customers using lateral flow assays, and plant-produced antibody, and customer tested it side by side with a traditional hybridoma-produced monoclonal antibody. In addition, I can't show, there's not enough time to show everything I want to show, but the differences in the glycosylation pattern was shown to have no impact on the monoclonal antibody activity within the diagnostic assay. We produced 3 batches with increasing levels of purification, and all behaved identically. And the customer stated that they could not distinguish between our plant-produced monoclonal antibody and their gold standard reference hybridoma-produced antibody. Next slide, please. Another good example of how our system can be used is for production of virus-like particles, especially in the given climate that we're in at the moment with COVID and needing to produce rapidly vaccines. So VLP vaccines can be prepared in around 12 weeks in the system that we use, our plant-based system, which is much faster than the traditional 9 months for the egg-based technology. So we have produced a robust copy mosaic virus, VLP, and we get very, very high yields for this protein. So we're looking at in excess of 800 milligrams per kilogram when we're purifying this protein. So yeah, there's a company in Canada called Medicargo, who are a much larger outfit than ourselves at Leaf, and they have produced VLP vaccines both for flu and for COVID, and the flu vaccine's been lodged with the FDA, and the COVID vaccine is under phase 3 clinical trials. So it shows that it's a viable way forward for producing like fast vaccines within the plant-based system. Next slide, please. So we can also produce cytokines and saponin. So these products are renowned to be quite difficult to produce, but they have benefits as therapeutic products. And we have actually been able to produce these products in our system. Next slide, please. So this is my final slide now. I'd just like to highlight that leveraging plant-based expression technology for healthcare improvement. So it's animal-free, cost-effective, and fast expression of drugs, diagnostics, and therapeutics for the human and animal markets. With a plant-based system, the cost savings can be used to ensure therapeutic products are affordable and can be used to improve healthcare throughout the world. In addition, it will produce products for low and middle income countries that might struggle to be able to afford the latest healthcare advances. So we have accelerated production times, therefore we could bring products to market sooner, saving and improving more lives, and also patent lives can be extended on potential drugs and products to bring to market, which is a big cost saving for the whole pharmaceutical market. Next slide, please. So thank you very much. And thank you to One Nucleus for inviting us to speak in this session.
Thanks, Leanne.
Thank you.
And thanks, Leanne. Great presentation. Some really exciting science. Can't believe that we haven't used plants before. It's just so sustainable. It's kind of links back to some of the topics we were talking about yesterday.
Yeah.
And so we have got time for a very, very quick question. There's a question from the audience asking about how this expression system, the yields compare to mammalian microbial platforms.
It really depends on the protein. It's on a protein, like I would love to sit here and tell you all that we can produce everything better than any other system, but as we know, biology is not a system that works like that. So it's sort of on some proteins will express much higher in the plant-based system than other proteins. We've got, I think on the catalogue, we've got ARA-H2, which is an allergen from peanut, which expresses at much higher levels within the plant-based system than it does in a bacterial-based system. We've had a client do some work on that. I can't share the data, but
sure.
Yeah, it's been shown that it does actually express much higher levels, and you get a more functionally active protein within the system as well than you would in bacteria. So
great. And just a quick one: are the plants harvested, or are they continuously grown?
No, we harvest them. We just harvest the leaves. We don't take anything else because it doesn't really. It just takes it up into the leaves. The stem isn't really as sort of permeable to the agrobacterium as the leaves are. It just takes straight up into the leaves.
Great, yeah. Thank you.
Thank you.
Thanks, Leanne. again, great presentation. So we're going to move on to our next speaker. This is Benjamina Bollag from Higher Stakes, and she's going to be talking to us about some of the work they're doing in the food industry. Over to you, Benjamina.
Thank you, Nadia. Could you bring up my slides, please? Amazing. Next slide, please. I'm Benjamina. I'm the founder and CEO of Higher Stakes. From the beginning of time, food has always been a way to show we cared. Feeding, nourishing, sustaining our families and communities. But more and more, we are realizing that food is also a medicine. The way we eat impacts our health in ways we are only just starting to comprehend. Our protein, fat, carbs, calorie system is so far from telling us how each bite that we take actually influences our health. But our direct health is not the only thing influenced by the food we eat. Livestock production can increase our health risk to the wider population, whether or not we are meat eaters. Next slide, please. Some of the many factors are respiratory diseases, zoonotic diseases, but probably one of the most concerning one is antibiotic resistance. There is many medically important antibiotics used on pigs in the US than on humans, and 30% of the world's antibiotics are used on pigs in China. This overuse and inappropriate use of antibiotics contributes to an increased resistance in microbes that could lead to as many deaths as cancer by 2050 if we continue on this trajectory. Next slide, please. Because of that, there's been a rise in alternative proteins. We've seen some of the world's biggest food companies launching new products, investing in this space. But most of those products are still plant-based meats. And if you're like me and still really, really love meat, this is not enough. Next slide, please. At Higher Stakes, we want to change the meat, not the behavior. We've created an extremely efficient and scalable system that allows us to scale any type of cultivated meat for us to continue to feed and care for meat eaters without harming our health. We take a small sample of cells from the animal, in our case, a pig, which we expand and then guide to become different types of tissues, such as muscle and fat. The advantage of doing things this way is that we have significantly less environmental impact, we don't need to use any antibiotics, and we don't hurt any animals. At Higher Stakes, we're working with a specific type of cell called induced pluripotent stem cells, which allows us to scale really rapidly without the use of fetal bovine serum and in suspension. We have a patent pending technology that allows us also to differentiate those cells extremely efficiently into any type of tissue without the use of genetic engineering, which allows us to go into regulatory approval more rapidly. Next slide, please. And this is not just a cool technology. This is also an enormous market. Within that market, we decided to focus on pork first. It's the most widely consumed meat and has enormous problems in the supply chain at the moment with African swine fever, killing millions of pigs. There's also the huge amount of antibiotic use, and it's genetically similar to human, which makes it sound gross, but that means that we can leverage more technologies from the therapeutic space, and vice versa, our technologies are more likely to be helpful in that space. Next slide, please. Well, at the moment, we're mostly leveraging technologies from traditional biotech. There's also a lot of technologies that we create that will be helpful beyond cultivated meat. Today, when the therapeutic space talk about large scale when it comes to induced pluripotent stem cell, we're talking 10 liters, 20 liters. But what we want to expand to, we're talking thousands and tens of thousands of liters so that we can create billions of kilograms of meat. There's more and more pressure on the biotech and pharmaceutical industry to lower the cost. And at the point where you have a small skin sample costing thousands and thousands, and we're creating kilograms at one pound per kilogram, there will be some questions. And at that point, people will start looking at how can we leverage some of the technologies that we created to lower the cost beyond that as well. While more and more companies in the biotech field are looking at the environmental impact, this is not at the core of what they do. But for us, this has to be one of our main focus, because our whole industry relies on us being more environmentally friendly. And so with that, we need to rethink some of the technologies that we're using. For example, growth factors that use a lot of water. We'll have to find ways of doing that better. Next slide, please. All of these challenges are not yet solved, and there are still both technical and non-technical challenges, like the regulatory pathways, that we need to address. But every problem is also an opportunity in disguise. These are opportunities for collaborations. These are opportunities for new startups to be created. These are opportunities for existing companies to reinvent themselves. And some of the areas where we're continuously looking to collaborate on are cell line development and banking, media development, scaffolding, bioreactor and facility development, automation, in-process monitoring, QA, and regulatory approval. And there are also many lessons that we can learn from parallel industry. I often get asked whether this is a race and whether this is a one winner wins it all industry. A good example is to look at the plant-based industry. There are many different approaches, and several of them have already proven to be successful, and many more will be. Impossible is taking the gene editing approach. Beyond Meat is taking the non-gene editing approach. Some companies are really not processing their meat and simply using a rare mushroom. Others are mixing it with real meat or using 3D printing. And there will be many more solutions. The barriers to enter are higher with cultivated meat, but there will be many different ways of tackling the cultivated meat sector. Some will work better than others, but there is room for many to succeed. One interesting example that you will all be more familiar with is the regenerative medicine space, where initial claims were grossly oversimplified and overplayed. There was fancy stories that resulted in unrealistic expectations and wild media hype. This peaked in the year 2000, where Time magazine named tissue engineers as the hottest job for the future. By the end of the year, 3,000 people had elected to pursue this hottest career option. There were 66 companies, some of them have gone public with public value over 2 billion, but by this age, the industry was starting to run into problems. The most serious of this was underperformance. The first products, although mostly approved by regulators, just did not behave as originally intended once they entered the clinic. Companies such as Appligraph broke down, merely acted as a wound stimulant rather than a living skin equivalent. And this is all despite investment in excess of 200 million. As if the scientific and operational challenges were not great enough, most of the tissue engineering companies were pursuing a biotech business model that was not appropriate for what they wanted. In hindsight, the dramatic decline of this field was inevitable, as it lacked the foundation of genuine scientific and commercial understanding. But despite this, and some of the research problems and corporate failures, the industry has gained back its momentum. So what can we learn? While we are all enthusiastic about the future of cultivated meat, we must remember that we are facing some very serious challenges, and that this is a field where we cannot move fast and break things, but have to carefully think about each different step, especially when it comes to the scale up. Going back even further than all this, the mid-19th century saw the development of a radically new direction in chemistry. Instead of simply analyzing existing molecules, chemists began to synthesize them, including molecules that did not exist in nature. It was unclear whether this field would lead, and many feared that its advances would lead to goals such as the creation of living beings. Today, nearly all aspects of our lives are touched by synthetic molecules that mankind has learned to make. Sounds familiar? We have gone through and will continue to go through similar changes. And I believe that cultivated meat, along with the whole cellular agriculture industry, is one of the most important ones of our century, with higher stakes at the forefront of it. Thank you very much. Next slide, please. No. Any questions? And we can maybe move to the next slide, please.
Benjamina, do you have another slide? Perfect.
Oh, yeah.
Great. Is that everything from your end, Benjamina?
Yes, thank you.
Perfect. Thank you. No, that's some really interesting work that's going on. I was wondering, have you had a sense of what the public's feeling towards eating meat grown in the lab is?
Yeah, so at the moment it's very positive. You see up to 70% of people that are willing to try cultivated meat. About 40% are willing to pay a premium for it as well. So at the moment it's very positive, which is really promising given that the technology hasn't even, reached market in most, most geographies.
Yeah, brilliant. And when do you think we'll see meat on the shelves that have been cultivated in a lab? Any timescales?
So it's already on the shelves in Singapore. So this is the only country that it's been approved. In Europe, the first applications are probably going to be by the end of this year, early next year. So we're looking at a timescale of 1.5 years minimum, because we go through the novel food regulation, the novel food regulations, which take a minimum of 1.5 years.
Great, thank you. Thanks, Benjamina. So we'll move on to our next speaker. So that's Sophie Vaud, who's a senior synthetic microbial biologist at Colorifix, and she's going to be talking to us about some of the work they're doing in producing novel dyes. Over to you, Sophie.
Thank you, Nadia, and thank you for the introduction. Could I have my slides, please? And thank you for the Helix committee to give me the chance today to talk about what we do at Colorifix in terms of sustainability and therapeutics. Here we are. Okay, no, I'm not Leaf. Colorifix. Okay, well, while the slides are up and running, I can tell you actually the idea of Colorifix and where it started. So 7 or 8 years ago, two of our founders, Jim Agioka and Orya Koni, were working on the development of arsenic biosensors for drinking water in Nepal. And when they went there, they realized actually that one of the largest sources of water contamination, next slide please, was actually coming from textile dyeing industry. And indeed, the dyeing industry is one of the most polluting sources around the globe. It consumes excessive amount of water, the equivalent of 30 million Olympic pools in a year. It destroys the local ecosystems, and it's detrimental to the health of the local population. So by seeing that, O and Jim decided that there was something to do and to leverage from the synthetic biology tools to find a natural, viable, and safe solution to make a change to this. Next slide, please. So Colorifix was founded then in 2016 and has put its roots down on the Norwich Research Park. Next slide, please. The mission of Colorifix is to develop a textile dyeing process that is more environmentally, socially, and economically sustainable. Next slide, please. Yeah. So our solution is a completely biological dyeing process to produce, fix, and dye with pigments onto textiles with absolutely no use of chemicals and between 60 and 90% of water consumption reduced and up to 70% of reduction of energy. Next slide, please. So what's behind our technology? So our technology starts with, the process starts with the identification of color or pigment pathways in any kingdom of life. Then we transfer these pathways into microorganisms that we can brew at scale in our proprietary bioreactors in situ, directly in dyeing houses, so that the dyeing liquor is then readily available to be used in a conventional industrial dyeing process to dye garments, fabrics, or yarns. Next slide, please. On the next slide, you're going to see examples of one of our latest colors and shades that we have produced at industrial scale. And you can, and I mean, you can't really see, maybe it's a bit small, but we are able to dye natural fibers like cotton or more synthetic fabrics like polyamide. And our process is actually virtually applicable to any textiles or any surface materials. And we are now in the process of expanding the color palette and increasing the scale of our production. Next slide, please. So what I'd like to do now is to bring you closer to our synthetic biology and microbiology process, which is at the heart of Colorifix activities. So our color development cycle can be assimilated to the systemic framework of the design, build, test, learn cycle. Next slide, please. So the initial design step is significantly supported by our computational biology team. They develop artificial intelligence-based retrosynthesis tools to identify potential pathways that can lead to the production of the pigment of interest, and they can also predict potential enzymes that compose these pathways and the associated genes or clusters that code for these enzymes. Once we have a genetic pathway candidate, we refactor it as to maximize its expression in our platform, microbial platforms of choice. Next slide, please. So to build this genetic construct, we have adopted the loop assembly system. The loop assembly system relies on type 2 restriction endonucleases, and it's a simple one-pot reaction that allows you to combine 4 DNA components into one receiver vector. And it works by levels. So you start at level 0. We have generated libraries of basic components, promoters, RBSs, coding domain sequences, and terminators. And you can easily assemble them in transcriptional units at level 1. And these units can then be combined together at level 2, 3, and so on into complex operands. And this system is great because level 0 parts are all standardized and they can be interchangeable. So it offers a great agility in the design of our genetic constructs. And it's also compatible with automation. So we're actually in the process of developing a high throughput assembly platform so as to speed up the assembly of our genetic constructs. Next slide, please. These genetic constructs are then transformed in a set of microorganisms, and we test them at increasing culture scales. We test them for microbial growth, but also for the production of pigments, and of course, the dyeing performance. At a screening stage, we are working with the Earlham Institute, also in Norwich, to leverage their, we work on a grant project to leverage their bioreactor microfermenter platform to rapidly screen media and growth conditions to optimize the production and the biomass production of our strains. And so this is very informative and is then further validated in our suite of lab scale reactors in our Norwich labs to characterize our strains and their metabolite profile using HPLC or spectrophotometer analytical tools. And then when we have a final strain, stable and everything, we can actually ferment it in Colorifix bioreactors that we have developed in-house and soon to be patented. And these reactors range between 30 and 3,000 liters of capacity. And as I said, alongside the microbial growth test, we also assess the dyeing performance of our strains. So we start at small scale on these multi-fabric swatches where each square is a different fabric. So we can quickly identify a combination of color and fabric that we are interested in before we work on the whole process to get a reproducible color at scale before we can actually produce in an industrial scale this color and dyes meters of fabric. And actually we are producing nowadays almost daily 200 liters of dyeing liquor, and soon we're going to increase this to 3,000 liters. So this is enough to dye about 500, 600 kilograms of fabric. So that's a lot of t-shirt actually. Could you bring me to the next slide, please? So then the last step of the cycle is the learn or optimization step. And in terms of synthetic biology, that means that we engineer the bacteria so that they are compatible with an industrial setting. We put also a lot of effort in elaborating our media recipe to include renewable feedstock, because we believe in sustainability and this meets our sustainability requirements. And it's also a way of keeping our process all cost effective and viable. And we also make sure that all of the media supplements we use are non-toxic to the cells, but also non-toxic for the environment. Next slide, please. So in terms of sustainability, alongside the conventional dyeing processes, we see more and more players that in the field that offer alternative or eco solutions. But what they do really is that they eliminate the use of petrochemicals to generate the dyes. But the rest of the process, unfortunately, is very similar to the conventional ones. So what we do at Colorifix is that we do not extract any pigment from the nature, but we rather get inspired by it and produce safe and optimized color-producing microorganisms that we can then readily ship to any client in the world into existing dyeing facilities, because we do not want to threaten the local economy and we don't want to delocalize or relocalize the dyeing activities. These fermenters are then producing in situ the dyeing liquor, and then alongside that we have developed a dyeing process that does not generate any toxic waste and works at low energy. Next slide, please. So alongside the sustainability has been actually demonstrated recently in our initial internal cost analysis on cotton dyeing. We have demonstrated that our process saves around 70% of water, steam, and electricity compared to a conventional dyeing, and produces 64% less of CO2. This is extremely encouraging for us. Next slide, please. And alongside the sustainability, next slide, please. We also want to produce a safe product. So we have tested cotton samples dyed with our pigments, and they all came back with the highest standard of safety in terms of toxicity, cytotoxicity, and they also have great fastness or resistance to light, water, washing, and rubbing. Next slide, please. We have also discovered that some of our dyes have yet more properties. So we have tested our red and one of our reds and one of our purple pigments for an antibacterial activity. And they proved extremely efficient in inhibiting 4 of the most common clinical pathogens, including Staphylococcus or Klebsiella. So these results, ironically enough, came back last March. So when the pandemic started and we've seen all of this scientific community coming together to fight the virus, we thought we had something to do as well. And so with 2 great teams of the University of Cambridge, we initiated the DAVIDYE project. So this project aims at developing antiviral dyes that are either mechanically interacting with the SARS-CoV-2 virus or generating reactive species that degrade the membrane of the virus, and all together, combined with our antibacterial dyes, we want to create a unique pigment formulation to apply on textiles that can be useful in the healthcare sector. Next slide, please. Next, yeah. So I would like to show you very quickly some very exciting preliminary results that we've got recently from the grants. So we have dyed some PPE materials with one of our pink in 2 conditions, pink A and pink B. And we have performed an antiviral ISO procedure using a SARS-CoV-2 surrogate. And you can see here on the graph that pink A and pink B have above 99% viral inactivation compared to the wild type non-pigmentin strain. So we are extremely happy with these results, and we are pursuing the development of a bacterial platform for production of a range of pigments with potential antiviral activities. We also want to understand the mechanism of action of these pigments in a molecular context. And I'm looking for more results on these grounds. Next slide, please. So yeah, here's the end of my presentation. I hope you enjoyed it. Thank you for your attention, and thank you for you see here all of our team, incredible people that make Colorifix definitely an exciting and colorful workplace to work at. Thank you.
Great, thanks, Sophie.
Thank you.
So we've got a couple of questions from the audience.
Yep.
So the first one is, can you see opportunities to make this process effectively carbon zero, and what would that take?
Well, carbon zero would be amazing. I don't think. So if you make carbon zero, that means you don't produce any CO2. So the moment we transport actually our strains, they're not going to be carbon zero. So we want to minimize as much as possible. And we are already actually working at reaching actually a lot better energy and water savings compared to conventional dyeing. So our objective, obviously ideal objective, is carbon zero. It's not going to be the case, but it's going to be a lot lower. And most importantly, it's going to be a lot less it's going to have a lot less impact on the health of the population and then the local ecosystems, which is also a great problem.
Great, thank you. And another one, how do you think textile industries can successfully implement a sustainable production process? What are the main limitations?
Well, I must say, actually, there is a change in the mentalities in the textile industry, a bit like organic farming 20, 30 years ago. Now there is willingness for change in textile industry. There are more and more labels that are driven by big companies and big brands to make a change and to have a more sustainable process altogether, from the production of cotton and fibers to the dyeing and then the distribution of the fabrics. So there is not so much pushback. Of course, our system relies on GMO, but there is no GMO released in the environment. Everything is contained. So I would say that across the board, we have a very good acceptance of our process.
Right. And it's great that your materials dyed with your novel dyes have got antimicrobial properties. Of course, it's very on trend at the moment, should I say, with masks. Do you see any other purpose that antimicrobial materials might be useful?
Any other applications?
Yeah, any other
yes.
application?
Well, of course, the textile is the easiest one, right? But as I said, our process can be applied to any surface materials. So you can think about anything like plastics, maybe.
Mm-hmm.
That anything that you can think about that is in the healthcare sector, like visors or anything.
Great.
It's a great application.
Thank you.
Thank you.
I think that's the end of our questions. Thanks to all our speakers. It's been a really great session. The thing I think takes out of it to me is there's lots going on in the life science sector, not just in healthcare industries, that we could really take home and use to make a better healthcare future. So I guess I urge everyone at conferences like on Helix to not only reach out to those healthcare partners that you might normally do, but also those smaller biotechs that have some really exciting innovations going on that we could probably use to produce a better healthcare future. So that's it from us. Thanks to everyone for joining us, and thanks again to the speakers, and I'll pass it over to Leanne again.
Thank you, Nadia.
Thank you,
And
Nadia.
thank you to all for the really good presentation. As you said, I'm particularly interested in hearing from these non-therapeutic life sciences, because I think the science is exciting, and also because maybe you're a bit less constrained with the business model of therapeutics biotechs, where you have more opportunities for being, yes, innovative, having some cash flow to fund the research, and I always find really exciting. And that's why I thought it would be interesting to present on that and see if there are any gaps that could fit into the therapeutic life science space. And as Nadia mentioned, I would definitely encourage this therapeutics life science space to look at that and think of bridges, collaborations, and others. So thank you for joining us today. I think it's a good starting point to try to mix a bit the crowd. And also on the geographical note, and that's linking with what we're discussing again this morning's presentation, having Norwich representation and also Cambridge representation is a really good show of what the East of England has to offer. So thank you for that. That was really good. So the session is coming to an end, and thank you again. And we're just taking a tiny break now, and we're back in, I think, 8 minutes, if I still know how to count. So at 11:30 for the next session on the future of drug discovery. Thank you very much.